streaming in live. I'm to online, that's what we say. I'm Tia Alan, and I'm here to take you through the process of fertilization in a flowering plant. Yeah, I'm here to take you through the process of fertilization in a flowering plant. And we said that for fertilization, the word fertilization means fusion. If I'm to repeat the content of the previous video, fertilization is the fusion of the male and the female gametes to form the male and the female gametes to form a zygote. Fusion of a male and a female gametes to form a zygote. And a gamete, for those who haven't covered this, a gamete is a reproductive cell. Is a reproductive cell. That's what we are saying. In plants, the male gamete is a pollen grain. And the female gamete are the egg cells found in the ovules. So for fertilization to occur or to take place, there must be fusion between the contents of the of the pollen grain and the contents of the egg cell or the contents of the ovule. This means that for us to continue, we must be aware about the structure of a pollen grain. A pollen grain is made up of an outer layer we call the exine, which is sculptured, rough, and tough. It is fully permeable to anything. It allows anything to pass through it. And then inside it is a small membrane we call a thin membrane we call the intine or the intine. The intine is surrounding the pollen cell cytoplasm in which two cells are embedded. Within the cell, the pollen cell nucleus, cytoplasm, there is the pollen tube nucleus. And another cell we call the generative cell. This is called a generative cell. Inside the generative cell, there is a nucleus we call the generative cell nucleus. So this is the structure of a pollen grain. Still, having known the structure of a pollen grain, you can memorize it there. You can look at it as we go on and memorize it, because it is going to be crucial in this process of fertilization. We must also know the structure of an ovule. An ovule is made up of it is made up of an outer coating that is double membraned, and this outer coating is known as the integument, that is made up of two layers, and then the point of attachment to the walls of the ovary. Inside the integuments, there is a nucleus. This space is a nucleus that is surrounding an embryo sac. It is surrounding an embryo sac. Above the nucleus, there is some space we call the chalaza, the cell chalaza. This is the embryo sac. These are the integuments. They are two layers. The integuments. We are looking at the structure of the ovule. And this small opening we talked of is the micropyle. The micropyle. Inside the embryo sac, there are cells at the opening near this micropyle. There are 3 cells, each with a nucleus, and the middle of which is known as the egg cell. These other 2 cells are known as the synagogues. In the middle of the embryo sac, there are 2 nuclei. And these 2 nuclei, or this pair of nuclei, they are known as the polar nuclei. Above which there are 2 cells. There are 3 cells. These cells, they are collectively known as the antipodal cells. So you must know the structure of an ovule. Majorly the egg cell, the antipodal cells, the micropyle. Let me hope we are following. So, yes? Yes, good. Invite your friend. Any question? We continue. Where is Labia? No, we are talking of the ovule and we are giving what we must know. Thanks, sir. That's great. Labia major. We are giving the key things we must know. Yes, they are part we are not including. There are many I'm not including. These are the key parts we must know in the process of fertilization, because they are the ones that involve in the process of fertilization. Any other question before we continue? Any other question? The embryo. I'm going to talk about the embryo in the process of fertilization. Thank you, Mr. Retire. Any other question, please, as we continue? Let's continue as they generate more questions. So in the process of fertilization, we must know this. If we remember very well, the carpel, or the female reproductive part of the flower, is made up of the stigma and the style that supports the stigma, and then the ovary down, in which the ovules are formed. The process of fertilization starts with a mature pollen grain landing onto a late and mature stigma. As the pollen grain lands on this, it absorbs water and mineral salts from the stigma. When this pollen grain absorbs the water and mineral salts, it germinates into what we call a pollen tube. It germinates into what we call a pollen tube. This pollen tube grows downwards towards the ovary, led by the pollen tube nucleus we talked about previously in the structure of the pollen grain. This pollen tube nucleus, behind it is lying what we call the generative nucleus we talked of in the structure of the pollen grain, if you remember very well. As this pollen tube is growing downwards towards the ovary, this generative nucleus divides by mitosis to form 2 meronuclei. If this is the pollen tube and this is the pollen tube nucleus at the tip of the pollen tube, the 2 meronuclei will come following the pollen tube nucleus. They are formed from the division of this generative nucleus that is lying behind the pollen tube nucleus in the pollen tube. Let me hope you are following as I continue. If you have any question, please pose it, I will answer it. A pollen tube, someone is asking what is a pollen tube? Like you've ever seen a stem. As a seed is, as the embryo of a seed is germinating into a seedling, it has a stem that grows fast. The pollen tube is also related to that, but this time it is the pollen grain that is germinating, forming a tube like a straw that carries the male nuclei to the ovules in the ovary. So the pollen tube is that tube that carries the male nuclei to the ovules. It grows down in the style towards the ovules in the ovary. So once the pollen tube is inside the ovules, let's take an example of one ovule here. Let's assume this is the ovule and the pollen tube has grown towards it like this. Once the pollen tube is inside the ovule, the tip of this pollen tube ruptures. It enters the ovule through the micropyle. The tip of this pollen tube ruptures. It bursts such that the generative nucleus disappears. Such that the pollen tube nucleus disappears. Once this pollen tube nucleus has disappeared, it will give room to the other two male nuclei to be exposed to the contents of the embryo sac of the ovule. Remember, we said that if this is the embryo sac, we have 3 cells at the opening of the embryo sac. One of the male nuclei will come and fuses with this egg cell to form what we call a diploid zygote. A diploid zygote. And then the second male nucleus, this was the first, the second male nucleus will come and fuses with these 2 polar nuclei to form what we call a triploid. Form what we call a triploid primary endosperm. The endosperm becomes, we shall look at the endosperm, what it becomes after fertilization. So, in short, the process of fertilization involves a rate and mature pollen grain landing onto a rate and mature stigma. Once there, the pollen grain absorbs water and mineral salts from the stigma such that it germinates into a pollen tube. The pollen tube grows downwards the style towards the ovary, led by the pollen tube nucleus. As the generative nucleus is lying behind, this is previously, the pollen tube has been formed after the germination of the pollen grain. The generative nucleus lies behind the pollen tube nucleus. As this pollen tube germinates, grows towards the ovary, the generative nucleus divides by mitosis to form, after mitosis. Mitosis means something dividing, a cell dividing into 2 equal halves. After the generative nucleus dividing by mitosis, it forms 2 male nuclei. So these are the 2 male nuclei that have been formed after the division of the generative nucleus. So as the pollen tube reaches or enters the ovule through the micropyle, this tip of this pollen tube ruptures or bursts. When it bursts, this pollen tube nucleus disappears. When it disappears, one of the male nuclei fuses with the egg cell to form a diploid zygote. Then, the second male nucleus that is remaining in the pollen tube fuses with the polar nuclei in the middle of the embryo sac to form what we call the triploid primary endosperm. We are looking at the process of fertilization in flowering plants. I will not write the content, but get the key points in this process. And the key points you've said: a right mature pollen grain lands on a mature right stigma. It absorbs water and mineral salts from the stigma such that it germinates into a pollen tube. The pollen tube grows downwards the style led by the pollen tube nucleus. The pollen tube, as the pollen tube grows downwards, the generative nucleus lying behind the pollen tube nucleus divides by mitosis to give two male nuclei. As the pollen tube reaches or enters the ovule through the micropyle, the tip of it ruptures such that the pollen tube nucleus disappears. And once this pollen tube nucleus disappears and this has ruptured, one of the male nuclei fuses with the egg cell to form a diploid zygote. And the second male nucleus that was formed from the generative nucleus division fuses with the two polar nuclei to form a triploid primary endosperm. And simply, that is the process of fertilization in a flowering plant.
That's more about pollen tube.
More about pollen tube, please. This is Tana. Tana. Even Red Zone asked about the pollen tube. That's great. We've said, I've been clear, I've said, a pollen tube, it is just as a passage that is formed from the germinating pollen grain that will deliver the nuclei from the pollen grain that is above the stigma to the ovules that are below. It is going to be that bridge that is formed from the germination of this pollen grain that will act as the passage of the nuclei, the male nuclei, from the pollen grain that is outside the stigma to the ovules that are down inside in the ovary. So the nuclei cannot move freely in the style. So they must pass through this bridge that is formed, which is the pollen tube that is formed from the pollen grain. That pollen tube grows inside this style, this tube here. When I break one from this, the pollen tube grows from inside here. I don't know if I can get it inside here. It grows inside this. I think you've ever seen flowers. There is a small tube that forms in a flower that has been fertilized. It acts as the passage for the nuclei, the male nuclei, from the pollen grain that is outside the stigma to the ovaries to the ovules that are inside the ovaries. So there's no much science about the pollen tube. It's just a tube or a bridge formed between between the pollen grain that is outside the stigma to the ovule that is inside the ovaries, acting as the passage for the two male nuclei for the process of fertilization to occur. You're welcome. Thank you, Tana. Any other question before we continue? Tana, thank you. Thank you, Tana. Any other question? That shows that you're following. That is great. And I'm so grateful with the turn up. Yeah, you're welcome. I'm so grateful for every comment we are making. So let's continue.
Nicholas is saying continue. Even Tana will continue.
Yeah, thank you. So there are events that occur after fertilization. That there are events that occur after fertilization. One, what occurs after the fertilization has taken place? I think we remember the diploid zygote that was formed. So the first thing that occurs, the diploid zygote undergoes development to form an embryo. We very well know embryo. Someone talked about the embryo. Excuse me. The embryo in a seed is that part that contains the primary and the radicle. If you remember very well, that's in your own information. Two, the triploid primary endosperm also develops to form a secondary endosperm that acts as a food reservoir in seeds. We are looking at the events that occur after fertilization has taken place. We've seen the process of fertilization. Please, if you can, rate a summary for this process of fertilization. Rate it based on this explanation we made. Know the structure of these ones, the pollen grain and the ovule. Don't make a detailed one, because I think the detailed one is for the advanced level. There are more parts we didn't include. We may not need at our level. And then we must know these events that occur after fertilization. We've said one, the zygote becomes or develops into an embryo. We are going to look at this in another topic we call growth and development. We very well know these points, these events. Three, the synagids, antipodal cells, the synagids and antipodal cells disintegrates. They disappear. We are looking at the events that occur after fertilization. Four, these are the technical ones we didn't see in our primary level. The ovules become seeds. Remember that. Five, the integuments become seed coats. We remember the events. the 6 ovary become the fruit, and the ovary walls become the pericarps of the fruit, and so many others. The calyx disappears or disintegrates, leaving a scar as one of the characteristics of fruits, and so many other things that occur after fertilization. We remember them in our primary, I'm very sure. So this is Mto Online. We said we give the primary information and make further research about the other needed information, and we give it for free. We deserve a subscription and that thumb facing up. Please comment. Any question before we continue? Any question? Do we have any question yet? And with that information, if there is no question, allow me break this session of fertilization so that we meet in fruits, if you don't mind, fruits and seeds. If you don't mind, should we do that? Great. Thank you. Selek Alan, Mto Online.
Yeah. Just enjoy the lesson. Kata Frank, well done.
You're welcome, Kata. Let's meet in the next.
Red Zone TV.